What if you need to quickly stitch random DNA pieces cheaply and without prep work? We know restriction cloning, Golden Gate, and Gibson are the standard methods, but let me introduce you to the rebels: SLiC, CPEC, and SLiCE. Total sequence independence. Let's go. These methods are different, yet share the same destination: assembling multiple DNA fragments in one shot. All three rely on 15 to 25 base pairs of homology at both ends. The only difference between them is the machinery used to stick those fragments together. Let's break them down. SLiC, or sequence and ligase independent cloning. It relies on the exonuclease activity of T4 DNA polymerase in the absence of DNTPs. By omitting DNTPs, you utilize the enzyme's 3'5' exonuclease activity to chew back the ends of your linearized fragments, generating the necessary single-stranded overhangs. By introducing a single limiting DNTP, in this example DATP, the T4 polymerase switches from exonuclease to polymerase, which immediately stalls due to the lack of other DNTPs, preserving the overhangs. Next is CPEC, or circular polymerase extension cloning. Forget the chewback enzymes. This is basically just a modified PCR that uses no external oligos. The homologous ends of your linearized fragments prime each other, assembling into a single circular molecule, all within a normal PCR cycling program. This entire assembly happens in only 10 to 15 cycles. This is often the sweet spot. And because it runs hot, CPEC can melt stubborn secondary structures or hairpins. But be careful. Do not overcycle the reaction and risk introducing PCR errors. And finally, SLiCE, seamless ligation cloning extract. This is the ultimate DIY method. It uses a cellular extract from E. coli, like DH10-beta derived strain, to do the heavy lifting ex vivo. Since you harvest the necessary enzymes yourself, it's insanely cheap once set up. The trick is that the lysate contains the cell's natural homologous recombination machinery, like the red ET system. This system seamlessly joins the overlapping ends of your fragments in one single step. Okay, before you run off, let's talk about what to watch out for. In reality, these are the usual problems you find in almost all cloning methods. Secondary structure. If your DNA ends forms tight structures, they don't anneal properly. This is less concerning for CPEC because its high temperatures melt those tricky structures. Repeat sequences. If you try to assemble genes that share the same, or very similar end sequence, you'll end up with frankenplasmids. Always use unique sequences for your overlaps. And that's the breakdown of SLiC, CPEC, and SLiCE. Remember, no single cloning method is best. Your choice depends entirely on the challenge. I've linked the original papers and protocols in the description below. Please like and subscribe. And let me know in the comments which method are you going to try first, or if you want me to explain any of these methods in more detail. Thanks for watching, and happy cloning.